Okay, hey guys, welcome back. I am
think you're talking to them.
I am here with Mia,
Hello.
and we are going to her internship. Do you want to explain what it is?
Yeah, so I can explain more inside, but I'm doing a genetic research study basically. So there's a certain gene with a variation, and I'll explain more of that inside, that is associated with bipolar disorder. So I'm seeing if that gene variation is more frequent in different ethnic groups or not, or if it's like the same.
That's really cool.
Basically, there's a gene called CACNA1C, and we're testing for a certain variation that's like highly associated with bipolar disorder. So people who have that variation in their gene tend to have bipolar disorder, and it's a single nucleotide polymorphism, SNP, which means it's just one nucleotide that's swapped. So like instead of, there's A, C, T, and G, instead of a G, there's an A. Did I get that? Is that the right? Okay. Yeah. So basically, we're running tests on that. So my project is to see if there's a different frequency of that variation in different ethnic groups. So I'm comparing Caucasians, Mexican Americans, and Korean Americans to see if there's if one group is more prone to that variation. Okay, so we have 7 samples. There's 230 Caucasians, 230-ish Mexican Americans, and then about 174 Korean Americans, and we're testing. So I just ran 7 trays with samples. Each tray is 96 samples on testing for one genetic variation, RS1006737, and then the second one we're now going to start testing for is RS, starts with 4. I don't really remember it,
Okay.
but that one's also highly associated with bipolar disorder. So we're testing for both of them. Right.
Hi, Petty.
Oh yeah, so I'll explain. So this is the mix we're doing. This is what's gonna basically bind to the strand if it has the variation. So yeah, in simple terms, I guess. But yeah, so this mixture will basically kind of help us identify whether or not the variation is in there, because this will bind to the gene if it does have the variation, and it will be picked up in the machine because it's runs fluorescent tests, and it can kind of pick this up. So because you'll see the master mix is pink, but we start with the cheap stuff first. So if we mess up, it's not expensive. So this is $3.90, and then these. So this is just Step C water. Okie dokie. That looks a little. Oh well. Sorry. Okay. And now we'll be adding the master mix. So that'll be 410 parts. To make sure it's actually in the liquid. Okay, perfect. Oh, sorry. I'm just gonna start taking the DNA out so they can thaw, but I have to make sure it lines up. So we start with 95. So this is, you put all the liquid in at once, and then you just click the button and it dispenses the right amount each time, so it's way faster. But you can only, unfortunately you can't do it for the DNA since they're all different, but when you're doing the same thing in each well, it's very convenient. Okay, so I just put it in. And then, okay.
And what have you set it to?
Oh, right. 9 microliters, because each well is going to have 10 microliters total. It's going to be 9 microliters of the mad mix, and then 1 microliter of DNA.
Okay.
In each one. Oh yeah, it's pretty straightforward. Very exciting. Oh, I forgot that those I wasn't filling. Okay, they all look.
Yeah, looks good.
Yep, that's full. Okay, thank you. So right now I'm just mixing it because we just thawed them, but we want to make sure it's really like there's no frozen chunks or anything. So I'm gonna do.
What is it?
So I have to go one by one, and I have to use a different pipe each time. So I just have to pipe one, pipe one, pipe one. But that's to make sure, because we really want to make sure that one person's DNA is in each tray.
Right.
And so this is, each number is just like a person. This is just like, I don't know, subject 95 or whatever system. So I have 3 ways to know that which one I'm on. So I can look at this one and see, okay, the first one, if I'm holding this one, I'm like, okay, I'm doing in the first well. I can also check here because the number of pipette tips that will be gone are the number I'm on. And I can check here because if there's 4 of these already in here, then I'm like, okay, I know I'm on 5 right now.
Right.
So that's what I'll be doing. First I have to, so this is the tiny little pipette because we're only doing 1 microliter.
Right.
I think this is what Dr. Kidd was asking me to explain. 1 microliter, and then so basically, start my first tip, get my DNA, and then so instead of just putting it in, I actually put it in and release 3 times. So this is how I mix it.
Right.
Okay, so this is the machine that we're putting our samples in to test. So basically, we already have the mixture that's gonna bind to the part of the gene, the CAC-NA1C gene, if it has a variation. But this machine kind of makes it so it can bind in the solution. So it's gonna, I think it like heats up so that the DNA separates into one strand DNA, and then I think it cools back down for it to attach, and then heats back up for it to make replications. And then the replication, because I mean, if it's just one tiny little piece, it's gonna be hard to see. So what this machine does is it heats up so that the solution that I put in can bind to the DNA and keep replicating until it's replicated enough that we can actually see it. And then this machine kind of shines fluorescence through the samples and picks up on the fluorescence if there is a genetic variation. So then this is the test right now. Oh God, this amplification looks really bad. Okay. All the other ones look nice and smooth. But so basically, so when it's done, it will show up here. So the U just means that we're testing for the the SNP RS1006737. And then here it's an N. This one we're expecting flat lines for all of them because it's just the mix. It's our controls. So like these 2 have no DNA. And then the rest will show up with different colored bubbles, either be blue, green, or red. Blue means wild type, and I'll explain what all this means. Blue means you're wild type because every person has 2 genes, one from their mom, one from their dad. So blue means both of them are wild type, which is normal. Yeah. And then green means you have 1 wild type and 1 variant. And then red is variant, meaning both of your genes have the variation. For wild type, let's say this is the RAM one. For wild type, you'll see that the RAM fluorescence steeps, because if they're both wild type, you're going to get a lot of this fluorescence because this is the fluorescent that binds on the normal gene that doesn't have a variation. And then if we're saying this color is the one testing for the variation, it's going to be flat. It's not going to be picked up because there is no variation, right? Whereas in someone, and then the control would just be also flat. And then in someone with 1 wild type and 1 variant, you're going to have, so the wild type one is going to go up because there's 1 wild type, and your variant one is also going to go up because you have one wild type and one variant, and then your control will be flat here. And then if you're variant, your wild type one will be flat. You'll have no wild type genes, and your variant one will be the only one that increases because only the fluorescence that detects the variation is picked up because there's no wild type. This one is wild type, meaning you have the 2 wild type genes, which means you don't have the variation that's associated with bipolar disorder.
Okay.
This one means you have 1 wild type, you have 1 normal gene, and then you have 1 gene that does have the variation. I want to say bipolar disorder is only associated when you have both variations. I think if you have 1 wild type and 1 variation, you're kind of good because maybe you're not like producing the right hormones because you have 1 varied gene, but you still have 1 normal gene that's kind of regulating still. And then this one, when you have variant, meaning both of your genes have the variation we're testing for, it's not, we can't prove that it always causes bipolar disorder, but there's a high association. Like it is mostly, if you have it, if you have this variation, 2 variations, you're very likely to have bipolar disorder. So that's what we're testing for.
Okay.
So this one was on our Mexican group. So you can see that the red is the people who were variant, and you see you have a lot of people who were wild type and 1 variant, and then these are the wild type.
Okay.
So the one we just did, which was, I'll pull up that one just because it's cool. So we had been reading articles that were saying that it's very, Koreans are very like the least prone to this variation, and you can see our tests are pretty consistent with that because the majority is all wild type, and even the wild type variant, you only have a couple. Looks like there's 5 of them, and then there's no variant. So far it's pretty consistent with what we were predicting, which is exciting.
Okay, so now we are going to lunch because Dr. Kidd has a meeting for 2 hours.
So we got a lot of free time.
Yeah. So, yeah, fun.
We are back from lunch, and this thing is finally finished. And again, there is no variance.
For the Koreans.
Yes.
Korean Americans.
So her hypothesis is looking pretty good.
strong.
Doing now.
It's very exciting. So I get to look at the graph, and I'm just going to copy 0 being wild type,
Yeah.
1 being wild type variant, and 2 being variant. I'm just going to put in the top right corner for each sample what they had, and then I'm going to enter that into a spreadsheet when I get home.
All right.
So
And what is the spread? What do you use the spreadsheet for?
to kind of calculate basically like how many variants we had.
Mia is doing some data recording, which you saw just now. Then she'll be filling trays, more trays. And then, looks like after that, the day will be over.
So you have 3 groups, and then you have a frequency of something you're looking for. So in our case, it's the frequency of the variation. So a test for homogeneity is basically testing to see whether the distribution of that variation is the same in all of the groups, or if the groups have like different distributions of the variation. So basically, our test was comparing first the frequency of the RS1006737 SNP, and seeing if all the groups had like a similar frequency of that variation, or if, you know, maybe one ethnic group, maybe like Mexicans had a lot more of those than Caucasians did, or Koreans or whatever. So we ran 2 tests, one for the first SNP, as I said, and then one for the second SNP, which was the 4765913 SNP. And both statistic tests gave us a p-value less than 0.001, meaning it's like really statistically significant that there is a difference in frequency of the variation between Mexicans, Caucasians, and Koreans, which basically is just really interesting because basically in my study we were able to conclude that there is a difference and that some groups are more prone to have this variation, it seems, and while we can't directly then conclude that a certain ethnic group will be more prone to bipolar disorder,
because this variation is associated with bipolar disorder but doesn't always mean you have it. But because of my research, with the conclusion we have, there's pretty likely chance that likelihood of having bipolar disorder also varies between Mexican Americans, Caucasians, and Korean Americans. So those were my findings.
Thanks for watching. It is Tummy Duckies, and don't forget to subscribe. Don't forget to like, comment what you want to see. I'll be doing a Q&A next week, so if you have any questions, leave them down below or DM me on Instagram, Snapchat, or even Twitter. And see you next time. Bye.